Hi, my name is Riti Sarangi. I am a senior scientist in the Structural Molecular Biology group at the Stanford Synchrotron Radiation Light Source, or SSRL. SSRL is a DOE Basic Energy Sciences user facility that also supports BER research. Here at SSRL and within the Structural Molecular Biology group, we offer X-ray spectroscopy capabilities to scientists who are conducting research in DOE BER mission science areas. X-ray spectroscopy is an umbrella term that includes several different characterization techniques, such as X-ray absorption spectroscopy, X-ray emission spectroscopy, and extended X-ray absorption fine structure, or XAFs. Now, for BER science, these techniques can be used in isolation or in a combinatorial fashion to obtain chemical and structural details about elements starting at phosphorus through heavier atomic numbers. Each of these X-ray spectroscopy techniques look at slightly different atomic phenomena and provide information about the elements bonding in the biological or environmental samples. This information ranges from their redox states to chemical properties and to molecular structure determination. And these molecular insights are important for building a mechanistic understanding of biological function at the mesoscale to system scale. X-ray spectroscopy is a powerful technique that can simultaneously provide both geometric and electronic information about the element under investigation, and you can do so in a single experimental analysis. So, starting with a complex biological or environmental sample, we can answer questions such as, is the element present predominantly as a mineral or as dissolved organic matter? Is it bonded to organic carbon or inorganic oxygen species? What is its redox state and how strongly is it bound in its chemical form? Questions like that. And because X-rays are strongly penetrating, they are superior at such analysis with the complex sample types that are typical in DOE BER research areas. So these techniques can routinely measure a diverse array of samples without extensive sample preparation, so no stains for the X-ray measurements that we do. Samples can range from dilute biological solutions and solids at tens of ppm or more to complex soil organic matter mixtures. This technique can be used to measure samples at elevated temperatures all the way down to 10 Kelvin. And in this way, X-ray spectroscopy is a strong complementary technique to others that are available in the structural biology portfolio. So what types of BER-related questions can be answered by the various X-ray spectroscopy techniques? In the past, we have applied these methods to look at the role of metal and nutrient, and in cases, contaminants in rhizosphere and plants. And the technique's ability to probe one or more elements in isolation or independently of other elements present in the sample make it a very powerful tool for chemical speciation. And so we can track minerals uptake and utilization in sediment-hosted microbial communities. We can probe how metalloenzymes are the enablers of catalytic reactions that uptake carbon dioxide or are involved in methane metabolism. We can also answer questions about trace elements and how their metabolism impacts biogeochemical systems and processes. In particular, we can trace the evolution of a particular element's redox state and bonding distribution under various stress or environmental conditions and see how that transforms over time. These are just some of the examples that may resonate with the DOE BER user community at large.